Hello everyone, welcome to EPG Pathashala. I am Dr. Vinuta, scientist at Division of Biochemistry, Indian Agricultural Research Institute, that is IARI, New Delhi, India. Today I am going to talk about regulation of enzyme activity. Under this module, we will be going to explore how to regulate enzymes by changing its activity. Regulation of an enzyme takes place depending upon the metabolic needs of the cells. For example, all the metabolic pathways will not operate simultaneously in a cell at any given point of time, like fatty acid synthesis, fatty acid oxidation, TCA cycle and other metabolic pathways, which operates based on metabolic demand from the cells and environmental conditions, uptake of nutrients from plants and intake of food from animals. The objectives of this module is to explore how to regulate enzymes by changing its amount or concentration and to explore how to regulate enzymes by changing its activity. The metabolic processes can be well coordinated in any complex living system is by regulating the activities of various enzymes, which coordinates, complements and regulate internal homeostasis of the cell. Being the mobile machineries, enzymes can be well regulated either by changing the amount of enzyme or changing the activity of enzyme. Let us see first how the change in the amount of enzyme concentration will changes the activity of an enzyme. Enzyme concentration can directly or indirectly meet the demands of biological system. There are different ways in which enzyme concentration can be regulated. The various ways are affecting the gene transcription, degradation of mRNA, post-translational changes, or concentration of substrate or coenzymes and metal ion activators. Let us see the first factor through which the enzyme concentration can be regulated, that is affecting gene transcription. The transcription of a gene into mRNA and further translating into protein or enzyme can be either modulated in a positive or negative manner. If it is modulated in a positive manner, it is called as a induction. If it is modulated in a negative manner, the process is called as a repression. These are the result of various extracellular factors like hormones, neurotransmitters, transcription factors, signaling molecules and calcium. Changes in terms of activation known as induction and the enzyme can be classified as constitutive enzymes and inducible enzymes. Constitutive enzymes are those enzymes which does not change in its concentration based on the inducers available, and in contrary, inducible enzymes change in its amount according to signaling molecules available in the cell. The second factor is degradation of mRNA. This is an example for negative regulation, or it is a negative regulation of gene repression. Repression causes a decrease in enzyme concentration and is manipulated by other molecules called repressors. Repressors act either before transcription or post-transcription for decreasing the enzyme load. Post-transcriptional gene silencing is a major cellular strategy mediated through small RNAs. Those are siRNAs, that is small interfering RNAs derived from microRNA molecules transcribed from cellular DNA can bind to specific sequences in the mRNA of a target enzyme. The resulting double-stranded RNA complex recruits an enzyme called dicer that cleaves the mRNA and thus decreasing the translational rate. Repression mechanism can also be known as feedback regulation, as they are usually the end products of biosynthetic reactions. De-repression is another phenomenon where the reversible removal of repressor or its exhaustion maintains the normal rate of enzyme synthesis. Ubiquitination and degradation is an ultimate mechanism where enzyme molecules are tagged with proteins called ubiquitin, which are later proteolytically degraded within the proteasome machinery. Thus, the concentration of enzyme is well maintained within the cell according to the metabolic need. The third factor is post-translational changes. Along with the translational regulation, post-translational regulation is also important to control the enzyme concentration. Some proteins are synthesized in its proactive form, which is later cleaved to get activated and released in a limited fashion as per requirement by chemicals or by certain proteases. Certain post-translational modifications focus on targeting to specific locations or fully get folded by various chaperones get activated by binding to other factors in the cell. To regulate the fully active protein, proteasome machinery take charge of the turnover process either through ubiquitination or through proteolytic degradation of lysosomes. The fourth factor is concentration of substrate, coenzymes and metal ions. The susceptibility of the enzyme to degradation depends on its tertiary active conformation. The presence of substrates, coenzymes or metal ion activators causes changes in the enzyme conformation and hence are capable to decrease its rate of degradation. Changing the amount of enzyme is a time consuming process and hence the changing activity rate of a pre-existing enzyme is the fastest way to modulate the system. There are various ways to regulate enzyme activity. They are allosteric regulation, feedback regulation, pro-enzymes or Zymogens, protein-protein interaction, substrate availability, enzyme conformation and pH, covalent modification, enzyme compartmentation, organ specialization and isoenzymes. Let us see the allosteric regulation of enzymes. Allosteric enzymes is formed of more than one protein subunit with two different sites. So, one of the site is called as a catalytic site and other site is called as a allosteric site. Substrate binding occurs at catalytic site and the effector molecules which regulate the activity bind to allosteric or regulatory site. An effector is positive if it enhances the enzyme activity. For example, ADP is an effector molecule for phosphofructokinase. An effector also acts as an inhibitor, then known as a negative effector, when it decreases the enzyme activity. Example, ATP and citrate are allosteric inhibitors in a TCA cycle. Let us see the mechanism of allosteric regulation. Both the catalytic as well as regulatory site change its shape or conformation. The two possible conformations which are in equilibrium exist. An allosteric positive modulator will bind selectively to regulatory site, which results in the binding energy to shift the equilibrium towards the best fit conformation. In a contrary, a negative modulator will selectively bind to allosteric and stabilize the enzyme inactive conformation. Allosteric enzymes shows a sigmoid plot when the velocity is plotted against substrate concentration and they do not follow Michaelis-Menten equation. The next factor is feedback regulation. Due to structural similarity between the products of enzyme catalyzed reaction with its starting reactants, the product can also bind to catalytic site with lower affinity. The product inhibition, or also called as a feedback inhibition, is a major type of regulation mainly based on high product concentration. This arose as an energetically advantage for a cell if the end product of an entire pathway would bind to initial enzyme, which results with the inhibition of whole pathway. Let us see the mechanism of feedback regulation. feedback regulation is different from feedback inhibition. So, what is feedback regulation? It means that an end product in the reaction decreases the rate of enzyme synthesis at the level of gene expression. It does not affect the enzyme activity. It decreases the enzyme quantity through the action on the gene that encodes the enzyme. It is a complicated process that takes hours to days. Example, inhibition of HMG-CoA reductase enzyme by dietary cholesterol. So, let us see what is feedback inhibition. Feedback inhibition of an early enzyme by the end product, and it will not affect enzyme quantity, and it reduces activity of an enzyme. It is rapid and direct, which occurs in seconds to minutes. For example, CTP inhibits ATPase enzyme in pyrimidine de novo biosynthesis. The other factor is pro-enzymes or Zymogens. Some enzymes like pepsinogen, trypsinogen, chymotrypsinogen, etc. are secreted as inactive forms called Zymogens or pro-enzymes. The presence of addition polypeptide which masks the active site makes Zymogen inactive. The removal of the inactive polypeptide converts this pro-enzyme to its active form. The activation of Zymogens can occur by any of the following ways, that is given in the slide. That is activation by HCl, activation by other enzymes, enterokinase, auto activation, that is enzyme activates itself. Let us see the biological importance of Zymogens. Some enzymes are secreted in zymogen form to protect the tissues of origin from auto-digestion. The another importance is to ensure rapid mobilization of enzyme activity at the time of needs in response to physiological demands. The other factor is protein-protein interaction. In enzymes that are formed from many of the protein subunits, the enzyme may be present in an inactive form through interaction between its protein subunits. The whole enzyme formed of regulatory and catalytic subunits is inactive. Activation of the enzyme occurs by separation of catalytic subunits from the regulatory subunits. For example, protein kinase A. The regulation of protein kinase A enzyme is through the protein interaction. This enzyme is formed of 4 subunits. 2 subunits are called as regulatory subunits and other 2 subunits are called as a catalytic subunits. The whole enzyme is inactive, that is 2 regulatory and 2 catalytic subunits. So, the CAMP activates the enzyme by binding to the 2 regulatory subunits, releasing 2 catalytic subunits and hence activating the protein kinase A enzyme. The fifth factor is substrate availability. Based on the structural similarity, reactants bind to enzyme active site with particular affinity and Km. If the substrate concentration is much higher than Km, signifies that the catalytic site got saturated and the enzyme is maximally active. While if the actual concentration of substrate in a cell is less than Km, the activity of enzyme is very low. The sixth factor is enzyme conformation and pH. Change in the hydrogen ion concentration due to active metabolic process like respiration also changes enzyme conformation and its activity. The initial changes are covalent due to change in the protonation state of the enzyme disrupt the delicate balance of force and result in structural change. The seventh factor is covalent modification. Post-translational modification can affect enzyme activity through local or global structural changes either by promoting or inhibiting binding interactions of substrate and its allosteric regulators. Changing the location is another post-translational modification. Certain modifications like phosphorylation, acetylation, methylation, sulfation, glycosylation, amidation, hydroxylation, prenylation and methylation which occurs in a temporary fashion also play part in enzyme regulation. Let us see some of the examples activated by phosphorylation. These are usually enzymes of degradative reactions. For example, glycosyl phosphorylase that breaks down glycogen into glucose, and another example is citrate lyase which breaks down citrate, and another example is lipase that hydrolyzes triglyceride into glycerol and 3 fatty acids. The eighth factor is enzyme compartmentation. Compartmentalization is also a type of enzyme regulation, localization to different environment from a reducing, that is cytoplasm, to an oxidizing, that is periplasm environment, or from high to low pH or high to low energy. Example, fatty acid synthesis takes place in cytoplasm, but oxidation of fatty acid takes place in mitochondria. The ninth factor is organ specialization. Each and every cell or tissues have a definite pattern of gene expression and different set of enzymes known as isoenzymes for specific set of reaction. Exokinase has another specialized isoenzyme form called glucokinase in liver and pancreas, which is more sensitive to glucose concentration. Glucokinase expressed in the liver and pancreas that has a lower affinity for glucose, it is more sensitive to glucose concentration. This enzyme is involved in sensing blood sugar and regulating insulin production. The 10th factor is isoenzymes. Different proteins which catalyze the same reaction are called as isoenzymes or isozymes. Usually, isozymes are multimeric proteins containing 2 or more different polypeptide chains. In isozymes, polypeptide chains are products of different genes and it catalyzes the same reaction can be located either in different places of one cell or in different tissues. For example is lactate dehydrogenase. To summarize, dear students, in this module, that is module number 13, we have discussed in detail on regulation of enzymes which depends on concentration of enzyme and activity of an enzyme.